is, right? Fine. So first, I would like to make you understand what exactly the secondary growth is, and what is the difference between the primary growth and secondary. I'm more than sure that you must have forgot whatever I have told you in the last class. Let me check with you. So what is the secondary growth? Can somebody tell me? No. Primary growth and secondary growth. Fine. So first, let me make you understand what exactly the secondary growth and primary growth is. But this time you need to remember it. Don't forget in next class. Look. Whenever you implant a small plant, so suppose that there is a small plant at your garden, right? And this small plant is, you can call it plant seedling as well. So this is a small plant. What will happen when you have implanted it? It was a seed-like structure. It was a seed-like structure. The seed germinated, came out of the ground, and small seedling is coming, and this small seedling is growing, right? I have already told you that what are the meristematic structure. I think it must be very clear to all of you, right? Now, what will happen when it will start growing? There are some meristematic tissues. These meristematic tissues are from since the birth of the plant, right? So these meristematic tissues, which have been formed from the since the birth of the plant, so we will call these meristematic tissue primary meristematic tissue. By the way, this is the apical meristematic tissue. So this is the primary apical meristematic tissue. Meristematic tissue. These primary apical meristematic tissue, they will increase the length of the plant. Again, at the tip of the root, I want your 100% focus over here. You won't get this thing anywhere, but I am delivering. These again, these are also apical tissue. We have already understood that these are also primary apical meristematic tissue. We know that meristematic tissue or meristematic cell are those cells or tissues which keep on dividing, right? Now, what will happen? These primary meristematic tissues, they will increase the, what they will increase? They will increase the height of the plant. They will increase the length of the root, right? So height or length, they will add on this one, right? This growth in increment of the height or length due to these primary tissues, primary meristematic tissue, we call it primary growth. So basically, the primary growth, what is the primary growth is, in general, if you'll ask me in the layman term, primary growth means increase in the length or height of the plant because of primary meristematic tissue. Is that clear to all of you? I think it should be crystal clear to all of you. Keep on responding me, please.
Yes, sir.
Yes, sir.
Now, what will happen? The plant which you have implanted in this rainy season, after 2 years or 3 years, this plant will not be any plant. Rather than this plant will become a huge tree or a big tree. So what will happen after some time? This plant will become a big tree. This plant will become a big. Now what happened? What exactly happened? Basically, the primary growth was increasing the height of the plant. But now what is happening? This stem was that much thick. It was a thin stem, but thin stem had become thick. Now what happened here? There were some tissues. Try to understand. There were some tissues which were permanent tissues. Now these permanent tissues, there were some permanent tissues. These permanent tissues have regained dividing property, right? They became secondary meristematic tissue. Why? Because they are not here since the birth. After the birth, at the adult time, right? They became the meristematic. They regain the capacity of. So we will call them secondary meristematic tissue. Meristematic tissue. These secondary meristematic tissue will divide and they will increase thickness or girth of stem. Girth of stem. What we call, because this growth have been formed by, or this growth have been generated by secondary meristematic tissue. So I will call it means increase in the thickness or girth of this stem. We will call it secondary growth. So increase in the thickness of the stem is called secondary growth. Is this concept is crystal clear that what is the primary growth and what is the secondary growth? I want answer from everyone.
Yes, sir.
Please note it down this at your notebook. Write, please note it down quickly, and then let me know. Then I will tell you how the secondary growth is taking place. Done? Note it down.
Yes, sir.
Okay. Shidika, done?
Yes,
Yashir?
sir.
Okay. Martin, done? Fine. So, okay. So now, how the secondary growth takes. Okay. The next one. Again, I want your 100% focus over here. Try to understand what I am trying to. Fine. Look. Now, this system. This system. I am going to tell you the anatomy of the system. This. So when we cut something like this way, when you cut something like this way, right? Like this way, in this direction, we called it transverse section, like you cut the cucumber, onion, and all that, right? So that is called transverse section. So this is the transverse section, right? TS. In short, we call biology TS, right? Now in a TS or transverse section, TS means transverse section. In transverse section of the system, you will see this is the stem. Inside the stem, there are vascular bundle. We know that in dicot, the vascular bundle are like this way. So these are the vascular bundle. These are vascular bundle. Between the vascular bundle, right, phloem is outside and xylem is inside, right? Phloem is outside. So let me write it down. This outside part, this shaded part, outside part, this part is basically phloem. And what kind of phloem? This is primary phloem because this phloem is present since the birth. So those tissues which are present since the birth, every time I am like we are saying them primary, right? So they are primary phloem. And inner side, the tissues which are present in inner side, they are called primary xylem, right? We have already discussed the xylem are meant for conducting of the water and phloem is meant for conduction of the wood. Phloem is meant for the conduction of food, right? Between the xylem and phloem, there are between the xylem and phloem means between these xylem and phloem tissue, there are some embedded or sandwich tissues which are meristematic tissues. They are called, right? They are meristematic. Means they can divide. Meristematic. These meristematic tissues are called intrafascicular cambium. So cambium is made up of meristematic tissue which have capability of division. Is it clear to everyone what is intrafascicular cambium? Intra, which is inside the what?
Yes,
Conducting. Clear? 100% clear? Yasir, clear? Ilam, is it clear?
Yes, sir.
Great, great. Sidika, are you getting it?
Yes, sir.
Yasir, are you getting it? Okay, I got your message. Fine, very good, excellent. If everybody is getting you, focus over here. Don't go anywhere. Right. Then again, between these tissues, there are this ground part. This ground part is made up of this complete ground part. This ground tissue is made up of permanent. Wait, permanent. What are those permanent tissues? Parenchyma. They do not have capacity of division. But at the time of secondary growth, try to understand me. At the time of secondary growth, these tissues regain these permanent tissues. They become meristematic tissue. I have told you, right? They become secondary meristematic tissue, and they complete this ring, right? So these the tissues, the meristematic tissue, which are formed at the time of, let me write, become meristematic at time of division. sorry, become meristematic at time of secondary. At the time of secondary growth. What we call them? We call them because they are present between two vascular bundles. So we call them inter. We call them interfascicular cambium. Interfascicular cambium. Guys, did you got my point? What is the interfascicular cambium and intrafascicular cambium? Got it?
Yes.
Okay.
Can I explain it?
Look, between two vascular bundle, initially the tissues were permanent tissues. Means they were made up of parenchyma tissue. They do not have capacity of division. When plant is planning to increase its girth, increase its thickness, right? At the time of when plant is planning to divide, plant is planning to get thick one, right? At that time, these permanent tissues, which I have shown with the shaded area, these permanent tissue become meristematic, right? And they make a complete ring. So these meristematic tissue between two vascular bundle, this is the first vascular bundle, this is the second vascular bundle. These meristematic tissue between the second vascular bundle, two vascular bundle, we call them interfascicular cambium. Clear? Is that clear, Martin? Now?
Yes, sir.
Okay. Again. Now again, what will happen? You will see at the time of secondary growth, so these are the vascular bundle. We have already understood that between the two vascular bundle, xylem and phloem, there are cambium, right? These are the cambium, right? Now what happened? This ring formation have taken, right? So interfascicular cambium and intrafascicular cambium, they have made a ring, right? This ring is called cambium ring. Cambium. So how the cambium ring have formed? Formation of cambium ring have taken place because of intra plus interfascicular cambium. Is that clear? Guys, is it clear?
Yes,
Cambium
sir.
ring?
Yes, sir.
Okay, I want you to please note it down this thing again, because this is the most important question of this chapter, and you most probably will face this question in your examination, and this will be full mark question, right? When you get the question, you have the make flow chart like this way, what I have made, right? Not what is in your NCERT or not what is in the PPT form, right? Please note it down, copy it as it is. Just copy and paste like I am doing, right? You have to do it in your notebook.
Done, sir.
Done? Okay. Fine. Done, everybody? Martin, done?
Yes,
Yasir?
sir.
Alia? Okay. Fine. So again, what will happen? Now, take one vascular bundle. I am taking this vascular bundle, right? This vascular. Take out this vascular bundle. What will happen? This is the vascular bundle, and there is a camera. This is outside, there is a primary phloem. And below it, there is a primary xylem. What will happen? This cambium, this is a meristematic cell. You know, this is the meristematic cell. What will happen? This is made up of some cell. Now this cell will divide, right? This cell will divide. So it will divide like this way, from toward the periphery, right? Periphery, right? What is the periphery? Meaning peripheral part. Here, this side and this side. What we call this kind of division? I have already told you. I think I know you must not be aware of that. What we call this kind of division when cell like this cell divide like this way? Tell me. Nobody will tell. So this is called periclinal division. This kind of division, when they divide, because they can divide like this way as well, like this way as well, right? So what will happen? There will be a meristematic cell, and it will add on a cell. Once it will add on cell, what will happen? Once it will add on cell like this, and once it will add on cell like this, right? So this cell have divided here. It have added one cell, and it will add a second cell. One cell, second cell. So primary phloem is already there. Try to understand. Primary phloem is already there. Now the division have taken place. Now the vascular bundle will become thick. Here a cell is added, a layer is added. Here again a layer is added. What will we call it? Right? We will call it secondary phloem. Right? Secondary phloem have been added on, and already the phloem which was present, we will call it primary phloem. So primary phloem will be pushed outside, and secondary phloem will push it outside. Again, lower side here, the cell which have been added, lower side here, the cell which have been added. What we will call it? We will call it secondary xylem, right? The secondary xylem have been added, and inner side, innermost toward the core side, this side, right? This one side, this side, inner side, right? So what we will call it? This is already which is present, that is the primary xylem. So primary, secondary phloem and secondary xylem will be added on. Try to understand. I will make one more diagram. Then you have to note it down, right? So by this way, what will happen? Now, two layers have been added. What are those layers? At the center side, the layer which is added, this layer will be what? Primary xylem have been added, right? Then outside to it, secondary xylem added, and outside to secondary xylem, one more ring has been added. This one more ring has been added. So this is called secondary phloem. Outside to secondary phloem, there is a primary phloem, and its center there is a primary xylem. Primary xylem. By this way, the new layers are being added on every time, and due to addition of these new layer, right, new primary, secondary phloem and secondary xylem, right, secondary phloem, addition of secondary phloem and secondary xylem, we call it secondary growth. And due to addition of these two layers, what will happen? Thickness of system will increase. Thickness of system will. Is it clear to all of you or not? Tell me. Crystal clear or not?
Yes, sir.
Please note it down as it is. Next step. Quickly.
Done, sir.
Done? Okay, great. So again, so now come to the PPT part. So when you get the question at your school, you have to do like this way, right? So I think you must have got this thing. There is a video. Let me share this video with you. By the way, you have got everything, but still, let me share this video. Look, this ring formation after this is the primary xylem. This is the primary phloem. Now there is a Between them, there is a cambium ring. This cambium ring is made up of meristematic cell. Secondary growth. How does the secondary growth take place in dicot stem? This is the cambium ring. It also have become thickened. This is the secondary xylem. This is the secondary phloem. Look, this is the actual diagram of microscope. Cambium ring. Look, primary phloem is outer side toward the bark. And what is primary xylem? When hold on here. What will happen? This primary xylem, when stem will become very thick, this primary xylem will get that much pressure, it will get crushed. So the primary xylem will get the crust that made the actually wood part. Got it? That make the actually wood part, right? And it become non-functional. And finally, you must have seen the black color structure at the center of these wooden log. So that black color structure where the resin have deposited, tannin have deposited, and the rest of the chemical organic material have deposited. That core part, which is black color part at the center of the log, that is made up of primary xylem, right? And outermost part, that is made up of primary phloem. And outside of the primary phloem, there is a bark, right? That bark which protect the primary phloem, right? Got it? Generally, I have seen a question, very common question, multiple times. This is called girdling experiment. If you make a ring-like cut around a stem of a tree, that much, 1 inch cut, what will happen? You have taken a very big knife, and outside of the stem, you have make a girdle-like structure, ring-like structure, that girdle-like structure. What will happen that? What will happen after a week? Tell me. What will happen after a week? Come on. Look, if you will make a ring, if you will make a cut, like this ring-like cut, right, inside, what will happen? You have broken the primary phloem and secondary phloem. What will happen? The organic food will not go inside to the root. Organic food will not reach to the root, and root will die. And you know, if root will die, plant will die. Got my point or not? Got it?
Yes, sir.
So this was the secondary growth. I think you must have got every point, right? And you have already got the points. And again, this is a diagram. So this is the diagram where you can see again xylem, phloem, and again the cambium ring formation have taken place, then primary phloem, primary xylem. You have made everything, right? So if you get the question at your school examination, you have to make this diagram, not that diagram you have seen. And I want you to please write it down. Okay. Do you want it to write it down? Or you will do it by yourself? Or do you want me to dictate it?
Dictate it, sir.
Okay. So put heading wise, subheading wise, right? Put the main heading, secondary growth. Secondary growth. Secondary growth in dicot stem, because in monocot stem, secondary growth do not take. Primary growth produces growth in length of primary growth produces growth in length of stem, root, and branches. Stem, root, and branches. Full stop. Next point.
So can you repeat the first point again?
Primary
Point
growth
one.
produces growth in length of stem, root, and branches. Full stop.
Yes.
Secondary growth is the formation of secondary tissues. Secondary growth is the formation of secondary tissues like secondary phloem, and secondary xylem. Full stop. In the nutshell, full stop. In the nutshell, we can say increase. In the nutshell, we can say increase in the diameter of a stem, increase in the diameter of a stem is called secondary growth. Next point. Next point. Put the heading. Formation of cambium ring. Formation of cambium ring. Formation of subheading. Formation of cambium ring. Intrafascicular cambium. Fascicular cambium are present. Intrafascicular cambium are present since the intrafascicular cambium are present since the starting as dicot stem. Intrafascicular cambium are present since starting as dicot stem have open vascular bundle. Full stop. The permanent tissue between two vascular bundle regain capacity of division, and become meristematic regain capacity of division and become meristematic. Full stop. They form interfascicular cambium. They form interfascicular cambium. Full stop. Next point in the same subheading. Both intra and interfascicular cambium, both intra and interfascicular cambium form a meristematic ring, called called form a meristematic ring called cambium ring. Next point. Next subheading. Next second subheading B. Addition of secondary vascular bundle. Addition of secondary vascular bundle. Second subheading B. Start writing. Cambium ring or cambium cells, cambium ring or cambium cells divide in periclinal fashion. Cambium ring or cambium cell divide in periclinal fashion. In periclinal fashion. Full stop. Due to division, they add on. Due to division, they add on secondary phloem at outer side. Due to division, they add on secondary phloem at outer side of ring and secondary xylem to inner side and secondary xylem to inner side of ring and secondary xylem to inner side of ring. Full stop. Next point. Due to addition of secondary xylem and phloem, comma, primary phloem is pushed outermost side primary phloem is pushed at outermost side and primary xylem and primary xylem and primary xylem is pushed to inner side. Full stop. Next, subheading C. Increase in girth. Increase in girth. Start writing down in subheading C. Due to addition of secondary phloem and xylem, due to addition of secondary phloem and xylem, thickness or girth of a stem will increase. Full stop. Trunk of a stem will become larger in diameter. Trunk of a stem will become larger diameter. This kind of growth is called secondary growth. This kind of growth is called secondary growth. Is that clear?
Yes, sir.
One last point, which is very important. Last point. The primary xylem do not get space at core. Primary xylem do not get space at core. It get crushed and become due to pressure. It get crushed due to pressure and it get crushed due to pressure as it do not get space. Full stop. Deposition of tannin, deposition of tannin and deposition of tannin and deposition of tannin and other alkaloid, deposition of tannin and other alkaloid takes place. Deposition of tannin and other alkaloid takes place. Tannin and other alkaloid takes place. That's how it makes wood. That's how it makes wood. Okay, clear? Is that clear?
Yes, sir.
So this was all about the secondary growth. And one more thing I want to add on here. Just let me see the heading. Secondary growth. Now, let me tell you what is the cambium. Right? What is the core? Look. So suppose that if my thickness will increase, if I will get fat, so what will happen? This shirt will get rupture because this shirt fits to me, right? If I will become more fat, this shirt will get tear. Will it happen or not? Tell me. Yes, Martin.
Yes, sir.
If you'll get fat, then your shirt will get torn. Clear? Is it natural phenomena? So, what is the next thing? That this shirt should also increase its space. It should be stretchable, but if it's not stretchable, then the size of the shirt will also increase. In the same way, around the stem there is a bark. If stem will get thick, you must have seen. That's why the rupture part of the bark you can see in trees, most of the trees, older trees. If the thickness will increase, thickness of stem will increase, then thickness of bark have also to be increased. Otherwise, the bark will get rupture. Are you getting my point or not?
Yes, sir.
So what is a cork? Cork is the bark of stem. Look, this is wood, right? Hardwood sort of this, right? This is the vascular cambium, and after the vascular cambium, there is a bark, right? There is a cork cambium, another cambium. This cork cambium will divide, and this cork cambium will add on again in the same way what happened in the tissue. What will happen? This cork cambium will add on the tissue at outer side and inner side, and that's how it will increase the thickness of bark, right? And this is called cork cambium, or this is formation of. Now, what will happen? This cork cambium. This is cork cambium. That is meristematic tissue, right? Right, and it will divide. Cork cambium will divide, and basically this cork cambium is known as phelogen. What will you call it? Phelogen. This phelogen, this layer is called phelogen, right? Cork cambium layer is called phelogen, right? So this phelogen or cork cambium, it will divide, and it will add on cell toward this side. So the cells which have this, it added on at this side. So these cells, they will make the cork, right? They will make cork. So this side they will make cork. Cork, like you must have seen in the bottle of champagne, right? So that is the cork. And this layer is called, this is called phelogen, right? And this layer is called phellum. This layer is called, this layer, the layer inside called pheloderm. Pheloderm. So inner side it will add on pheloderm, another layer, and outer side is this layer, right? Outer side. So inner side it will add on pheloderm, and outer side it will add on another layer, and that is called phellum. Phellum. Phellum will make cork. The same cork which is used in the champagne bottle and all. Before the invention of this cap of the bottle, before the invention of this cap of the bottle, you must have seen in the older time, the cork is used in place of this cap, and those cork is basically made up of phellum. Is it clear or not?
Yes, sir.
Martin, Shridika, Yashir, is it
Ye-
100%
sir.
clear to all of you?
Yes, sir.
Very good. Excellent. Uh, so now make this one only. This is structure only, and then I will make you note it down. Fine. Note it down. Done.
Yes, sir.
Yes, sir.
In order, note down for cambium. In order to, what should be the heading? For cambium, yes, for cambium. In order to prevent, in order to prevent rupturing of bark, or in order to prevent rupturing of bark or outer ground tissue, rupturing of bark or outer ground tissue, formation of cork cambium, formation of cork cambium or phelogen, formation of cork cambium or phelogen takes place in dicot stem, and dicot stem. Full stop. Generally, it is formed by generally it is formed by hypodermal. Generally, it is formed by means below epidermis. Generally, it is formed by hypodermal tissues. Next point. Phelogen divide on both sides. Phelogen divide on both sides by periclinal division. Full stop. The secondary tissue produced inner side are called phyloderm. Comma, full stop. They are secondary cortex tissues. They are secondary cortex tissues. Full stop. Phelogen add on phellum or cork to outer side. Full stop. It consists of dead and compactly arranged cells. Dead and compactly arranged cells. Full stop. The cork cell contain tannin. The cork cell contain tannin. Hence, the cork cell contain tannin. Hence, they appear brown in color. I think it should be clear to all of you. Clear?
Yes, sir.
Yes, sir.
Great. Alia, is that clear to you?
Yes, sir.
Very good. Martin, Yasir. So tell me, let me tell you again, every time I am emphasizing on this thing, because this is really very important. So these both core cambium and secondary growth are the most important point. And I think that near school examination, the long question, it comes from this chapter, definitely it should be taken from. Some small things are left. What are the hardwood? What is the springwood and hardwood, right? And springwood and autumnwood, right? Hardwood and sapwood. So, at every season, what happens? In winter season, plant require less amount of the water, right? So the lumen or the diameter of the xylem will be very narrow, because they require. So if the diameter of the xylem will be narrow, the wood will be more solid, right? And again, what happens? So in spring season, the cambium activity. The vessels will be. So what will happen in spring season? The xylem element or the vessel will have wider cavity. The wood which will be formed in this season will be called spring wood, and again the wood which will be formed in the autumn time, right? So that will have narrow cavity wood will be solid. That will be called date, right? Or autumn. Got it? What is the spring wood and autumn wood? So let me write it down. Then you will get more a better idea. So spring season, the plant will require more water. Will it require more water or not? More water
Yes, sir.
because high transpiration. Then xylem will have wide lumen. So what will happen? Light color wood and this will be called, you can see over here, this is autumn wood and this is spring wood, right? So this is called spring wood, right? It will be less strong, right? Then what is the autumn wood? They require less water. Why? Due to less transpiration. Then what will happen? Xylem will have narrow lumen. Then dark or more density of wood. It will be a bit stronger. This is called autumn wood. It will be stronger. Got it or not? Very logical kind of. Clear? So please note it down.
Yes.
Spring wood and autumn wood.
Yes, sir.
Note it down. Spring wood and autumn wood. Copy from here. Easy. Let me know when you note it down.
Done, sir.
Done? Okay. One more thing, last but not the least. What is heartwood and sapwood? Look, So this primary xylem, this part, this primary xylem, this part. This is basically hardwood. Hard, right? Because this is a stronger one. Why? Because this is completely dead due to deposition of tannin and all that, right? This is darker in color, right? Dead and dark in color, right? And this part, which is made up of wood, is made up of xylem only, right? Then this is called sapwood. Why we call it sapwood? Because this is light in color and less strong. Because this is functional part, right? So that is called hardwood and sapwood. Please note it down. The region which is comprises of dead and the region which is comprises of dead and highly lignified tissue is called hardwood. It is non-functional. And do not conduct water. It is non-functional and do not conduct water. Next point. The peripheral part, which is functional and made up of secondary xylem, it is lighter in color, called sapwood. Hardwood and sapwood can be 2-marker question in your school exam. That's it from for this chapter. That's it from my side. If you have any question, please ask me. We have completed this chapter finally, right? In the next class onward, I will start from the structural organization in animal. Basically, this is animal anatomy part that will start. Tell me, is that clear?
Yes, sir.
Great. Okay. If you have any question, you can ask me. Else, bye-bye. Take care. Good night. See you next class. And please go at your home and revise it. You're already at your home. Revise it properly and